Hello everyone and welcome to another video from Bio-ProTech. So in this particular channel, as you know, that we intend to provide you information for free on the tangents of your bioinformatics, AI, and the solutions of your AI and computational part, which can be collaborated with your life science wet lab based experiment, and how to analyze those particular data sets, okay, that we see. Okay, and apart from that, we will also cater to many topics in the coming up sessions, from the likes of your how to analyze NGS data, then how to come up with the drug discovery experiments, okay, then what are the things that you can do using visualization of visualization libraries like your Matplotlib, this, that, okay. So all of these things would be covered in the channel in the upcoming videos, okay. So stay tuned, and if you haven't subscribed it yet, so please hit the subscribe button and like and share the videos with your peer ones. Okay, so with that particular note, this is myself, Pradyut Banerjee. I am a bioinformatics and CADD based scientist at a Bangalore based firm. With that particular note, so let's get started with today's video. So basically today's video topic is very intriguing for many of you. So this is on your computer aided drug designing. So as I have heard of this particular query that most of you are interested in this particular topic of drug discovery and designing, and this is my domain expertise as well, so I will just guide you through that. So we already have a video on that, okay. So that is being posted 2 or 3 weeks back, okay. So you can check that out. That is on like how to use AI or bioinformatics based tool for your molecular docking. So molecular docking is a part of drug discovery, but it is not the whole drug discovery, as you understand, because docking is about like interaction of the molecule, okay. But when we are talking about the interaction between the drug and the protein molecule or the receptor, many things comes before that also, that particular drug molecule is having what kind of a pharmacokinetic and pharmacodynamic profile, because we need to understand that the drug molecule or the ligand, or basically it is a chemical molecule, let's suppose, okay. So it can come from many sources. It can be from a medicinal plant, like suppose phytochemical, again chemical molecule only. It can be a synthetic chemical that is synthesized in your labs, okay. And it can also come from like repurposed drug. The drugs are which are already available in the market are already FDA approved or CDSCO approved in India, basically, okay. So after that, so after a decade of their use for a particular purpose, they are serving a particular purpose, but similar purpose we can search for that, okay. So like similar or different kind of disorders that can be treated with that particular drug use that we can find, okay. So that is called as a repurposing, because we are changing the purpose of the drug, basically, okay. So that is there, okay. So this can be the majorly three sources of your lead molecule, which later on might be converted to your ligand, which can be acting as an inhibitor or a substrate against your like receptor, that would be protein or macromolecule in this particular case, okay. So but before that, we need to ask us many questions, okay. So whatever chemical molecule, let's suppose there are a whole lot of chemical molecules relevant in this particular world, okay. But all of them cannot be administered inside our body, because that might generate a immune response. So that is the first thing. Second thing, how safe it is to administer inside the body. So will it cause any harmful effect in our, what do you say, organs, important organs? Or let's suppose it is causing carcinogenicity, it is having carcinogenicity, basically causing cancer, okay. Or many of such things, like your it is having higher nephrotoxicity, neurotoxicity, that comes under pharmacodynamics, actually, okay. What the basically your drug does to the body, okay. Whereas what body does to the drug, that is your pharmacokinetics, okay. So under which comes your ADME, okay, absorption, distribution, metabolism, and excretion, okay. And the toxicity comes under your pharmacodynamics, okay. So that is the basic difference. If you are from pharma background, you will understand that, and if you don't know, now you know it, okay. So basically the first thing that we are going to ask is not even this particular question. The first question that we need to ask is the molecule druggable, yes or not? Druggable is what? Druggable or drug, like it is having drug likeness, high drug likeness or not? Drug likeness basically is a parameter, so that defines that the particular molecule can be considered as a drug inside the body, so that it does not generate immune response, so it does not treat it as a foreign compound and like have some hyperreactivity reactions. Apart from that, there are certain parameters like it has to be within this particular molecular weight to particular like pass this particular thing. Apart from that, how many hydrogen bond acceptor, donor, all these things are there. Okay, so for that we have many rules. Okay, like your Lipinski's rule is the golden rule of thumb, actually it is called as. Okay, so Lipinski's rule of five, it was called as. Now the five rules are not there, four rules are actually there now. Then you have Weber rule is there, Ghosh rule is there. Then you have your big pharmaceutical companies also have their own rule, like your from the likes of your Pfizer, GSK, okay, Golden Triangle, Pains. Okay, so there are a whole lot of rules. Okay, so you can basically try to satisfy four or five of them that will do the job. Okay, and once we are done with the drug likeness, that we are confident enough that the particular compound can be a drug, post that we will move on to the pharmacokinetic and pharmacodynamic property check, that is your ADMET. Okay, so then after that we'll be moving on to a molecular docking. The video is already there, you can check it out. So molecular docking will check that because the drug, when it is absorbed well, metabolized well, then you are distributed in the blood well, then it will get to that particular point of contact to the receptor. No, so then comes the question of docking. Before that, it doesn't make sense that we are checking the affinity score, this, that. Okay, if it doesn't get to that particular place, how can we ask this particular question? How will it interact? Isn't it? So likewise, so first the docking will happen, and post that we can see that what is the affinity score, how it is interacting, what kind of bonds it is supported using. Then comes a post-doc analysis phase. Okay, so where we'll like understand what kind of like bonds it is supported using, how many covalent bonds, how many like what do you say, your hydrogen bonds, how many hydrogen bonds, basically what is the bond length of that. Shorter the bond length, obviously stronger the bond is. Okay, and the strength is. After that, we will move on to your validation. In silico we can do the validation via the means of your molecular dynamic simulation, so which will take that particular thing, and like simulation means to mimic human body condition. So you might be having a question, we are like designing the drug in computer, how will it work in actual life, how do we know? So there are two ways to know that. One is obviously wet lab based experimental validation, that is possible. Okay, so that I'm obviously not going on to, but I'm telling you about the in silico based validation technique, that is your molecular dynamic simulation, where we simulate all of the human body condition that the receptor is experiencing, as well as the ligand when administered inside the body, that will also experience the same. And it will give us an idea that for, let's suppose, for like 100 nanosecond or 1 millisecond, so this particular interaction is happening in the 3D space in docking, we have seen that. But inside the human body you have water, you have what do you say, temperature, pressure. So we need to basically ensemble all of those. Okay, and provide that particular similar condition that a ligand or a drug molecule will face inside the body. Okay, so in that particular way we can know that the interaction is happening for a longer duration, and is it moving away from the cavity, like is it denaturing the protein or whatever. So there can be multiple circumstances that can follow. Okay, and then comes the analysis of MDS results from the likes of your like RMSD, RMSF. Okay, radius of gyration, this, that. So I will make a dedicated video on MDS simulation because that is a subject by itself. Okay, so that cannot be covered in one video. Okay, so in today's video, basically I will give you a brief on how to do the drug likeness screening, how to do the ADMET screening. Okay, so obviously we start with a huge library of leads. Okay, so here I will only take only one molecule and show you how to do it, and like that needs to be done for whole lot of molecule. Okay, lakhs of compounds that you do it for, and then you screen based on that. Obviously the screening method is a elaborate one that we cannot cover over here, but I will show you the way how to get your drug likeness and ADMET check done. Okay, for that we'll be utilizing two tools majorly. One is your SwissADME, that is very much famous. Okay, apart from that, we will also use a ADMETlab 3.0, ADMETlab 3.0. So that is my favorite tool. I would recommend you that because that has more of such parameters, and obviously both of them are backed by your Python. So basically it like extract the data from like using Python only. Okay, so likewise, okay, so you will see that what kind of parameters we are getting under all of them, and these two softwares are capable enough to do your drug likeness screening as well. So only one software you need for all of your drug likeness, ADMET, everything. Okay, and post that, once you are done with that, you have defined the lead, or you have finalized the lead or chemical molecule that you want to proceed as a drug. Okay, so then you have to check the like affinity score of that using docking, and further after that you have to do the simulation and likewise. Okay, after that also you have multiple things going on, but I will just restrict the video to this particular 4 steps that are very major. Okay, and that is what you need to understand as a beginner. And if you are at advanced level also, this might also help you like coming up with further part of the things. Okay, so with that particular note, so let's get started. Okay, so I will just show you the workflow once, and then post that we will directly move on to the practical demonstration. Okay, so here we are. So basically, you can see this, I guess. So this is computer aided drug designing pipeline, as you see. Okay, so here it is. So you can see this particular first things that is called as your drug likeness. Okay, or also called as your druggability. Same thing. Okay, so you can use, I have already stated Pfizer, GSK, Weber rule. You can see Ghosh rule is there. Lipinski's rule of 5 is the golden rule out there. Okay, then QED is also there. Okay, apart from that, after doing the drug likeness of a particular what do you say, chemical molecule, so then you will go on to your ADMET. So that is your absorption, distribution, metabolism, excretion. All of this particular 4 falls under your pharmacokinetics. Okay, then the last one is called as your toxicity, that falls under your pharmacodynamics. Okay, so those screening are to be done. So as you can see, so we will be testing for your obviously toxicity results. Okay, metabolism, how it is metabolized, is it causing some harm to the liver or not? Okay, so is it how like toxic to the kidneys or not? Is it causing any genotoxicity, then your nephrotoxicity, then your autotoxicity, eyes basically. Then the multiple types of oral route toxicity, that because most of the drug you will be taking orally only, so is it having any oral route toxicity or not? Okay, so those things. So then solubility of it, then permeability of that particular thing, so all of these things would be judged upon. Okay, then after that we'll move on to the docking. So you can see a lock and key kind of a model. Obviously docking works like that only, lock and key kind of a thing. Then obviously binding affinity, what do you say, more it is in the negative connotation, better it is. But obviously with some exceptions like diffdoc base tool, diffdoc basically, in that a higher positive score is a better score. That particular part is covered in the video, so you can go through that. Okay, so I will just attach that particular video at the end of this particular video as well. Okay, so docking, so using your SwissADME and diffdoc. So binding affinity interaction analysis can be done using PyMol. So that particular video is also there. Okay, so the PyMol video is also there, that after binding affinity, after getting the docking score, how to get the interaction done. Okay, so that particular video is also there. Like only 1 week back, you can find it. Okay, so you have, then post that you have your molecular dynamic simulation. I'm planning to come up with a dedicated video for this as well, because this is a subject by itself. So you have your like a molecular dynamic simulation for your protein ligand interaction obviously, but here we will be taking all of the practical things into account, and we are trying to simulate the human like conditions. Okay, so that is the main purpose of a simulation. That is how simulation works actually. And as we are taking the molecular dynamic changes into account, basically the dynamics means what, because the molecules are not stable, it will be rotating about its axis, along the axis. Okay, so it would seem vibrating to you. Okay, so you will see that, but actually what is happening inside the body, obviously all the molecules are in tension because of the like energy differences. Okay, so like pressure, temperature, many things are there. Okay, so we'll talk about that. Energy minimization is also a step in your dynamic simulation, likewise in your docking. Okay, so there are much more steps in here. Okay, so likewise, and for that, for pursuing a good amount of simulation or good like a time bound simulation, you need a supercomputer or HPCC or high performance computing clusters in most of the time. Okay, so that is there. So here in today's session, we'll be talking about your drug likeness and ADMET screening. Okay, so I will just take one chemical molecule and show you across both of the things. I will show you using two tools, basically your SwissADME and ADMETlab. So let's get started with that. So you just need to open your regular search engine, whatever search engine you are using. Okay, so and you go to your search engine and simply type in. So first I will demonstrate using SwissADME, but prior to that we need to take a smile formula of any of the chemical molecule. Okay, so obviously when we are talking about your library of molecules, so there will be lakhs of molecules, thousands or lakhs of molecules. Each time you have to do it for all of them. Okay, so I'm just showing you one of them, like using one of them, and the process remains the same. Okay, so it is no different. So let's you can go to PubChem, Zinc, or Drug Bank, or any Bitviz is there, Hugging Face is there. So there are a whole lot of tools to search your chemical structure from. Okay, so you just go to this, then you have your go inside PubChem, and let's like you can type in some like a name of any of the chemical molecule that you want to type. Like a phytochemical search, if you are doing, you can do it from IMPAB, Dr. Duke's, Coconut database. Okay, so likewise, apart from that, if you are like going for a synthetic chemical, let's take a already published or what do you say, already marketed drug for that instance. Okay, so aspirin, as you know, it is used as a like analgesic as well as your what do you say, your like it is also used as a blood thinner, and it has a multiple, what do you say, use cases. Okay, apart from that, so it is being studied, so I'm just telling you like uh, from here only that it is having a toxicity profile, mostly not very high toxicity, but it is having a liver injury profile. Okay, so that has been studied also. You can go through the research papers, that multiple research papers are published, that it has a bit of a higher liver, what do you say, liver injury causing causing it can do. Okay, so that particular thing is there, so we will check that out. Okay, so while we are checking for ADMET, obviously in under toxicity we can see that particular parameter is it having that particular thing or not. Okay, so it is not only with the published drug, you can, or basically for any ligand molecule it will work the same. Okay, so here you can see, yeah. So I can basically choose like your stick or ball, that doesn't matter, because I'm not going to download anyways the 3D structure over here, because that is not required. For docking we require the 3D structure. Here the 3D structure is not relevant. Okay, so we only require the formula, and the simplest formula that is like worldwide recognized, that is your smile. Okay, also called as the canonical smiles. Okay, so canonical smile formula, this is the simplest formula that you just copy. Okay, control C also you can do, or command C, whatever system you are using, works the same. So now let's go on to your uh, the tool. Okay, so that is your SwissADME. It is from the Swiss Institute of Bioinformatics. Okay, so so that is from Switzerland. Okay, and that is a very what do you say, good company, having all of the drug discovery and tools likewise. Okay, so I have also demonstrated SwissDoc, which is from the same suit. Okay, from the same company basically. So let's go inside SwissADME. Okay, so here you can see that like all of the tools are enlisted over here only. SwissDoc, this is the SwissDoc that we have used for our docking purposes initially. Okay, so here we have your SwissADME. Okay, Swiss target prediction is also there. Okay, so in order to predict your target, so you can attach actually multiple smiles over here, but as we have come up with one of the smiles as of now, so we will like paste the smile over here. Okay, and if you don't have the structure, you want to construct a structure, so the Marvin JS is also having a collaboration with them, so you can paste the structure over here, and Marvin JS will basically create the structure for you. Apart from that, if you want to do it manually, you can select the benzene ring and then the carbons and this that. Okay, and then make your own structure. You just have to satisfy the balances and all that you already know. Okay, so here there is a run button. Okay, so you can't see because of this, so you can like open it and see it by yourself. Okay, so I can hit on run and it is already running, and because this is just only one molecule that I have attached, so it would be very fast. You can also go by example. I will also show you via that. Okay, so you have the formula of it, molecular weight, this that basic information. Apart from that, lyophility. Okay, so lyophility information is given over here. Okay, what is the log p value? Okay, so so the solubility part is also covered over here. You can see the water solubility. Okay, water solubility is there. Okay, Apart from that, this is your pharmacokinetic studies, ADME basically. So here you can see what would be the absorption, what would be the blood brain barrier permeability, permeability. Okay, so yes or no. Apart from that, these are the metabolism parameters basically. Okay, cytochrome P53 parameters this are, CYP1A2 inhibitor or substrate it will work as. Okay, so the substrates are not enlisted over here, but you can find those in your ADMETlab. So that is there. So obviously there is a boon and bane of all of the tools that you see. Okay, so then you have your log KP. Okay, skin permeability, it gives an account for. And if you want to learn more about it, you can point your cursor towards the question mark and click on that also. Okay, so it will give you the idea of what is this particular parameter all about and what it is counting upon. Here you can see solubility should be less than -10. Okay, or likewise pour. What would be the empirical decision? Okay, so that you can go by. Then you have your drug likeness. Okay, so the drug likeness features like your Lipinski's is there, Ghost rule, Weber, again. Okay, so likewise many rules that you can see over here. Okay, apart from that, your pains rule is there. Okay, so pains, then drug likeness, multiple rules you can find. Apart from that, if you want to have a representation of this particular permeability, and likewise you have one more thing that is called as a boiled egg model. Here you can see, after you boil an egg, you have the egg yolk and the egg whites. Okay, so this is the egg white and this is the egg yolk. Okay, so anything which falls under your egg yolk part, okay, so that will have a good blood brain barrier permeability. As you can see, good blood brain barrier permeability it would be having. HIA would be high if it is falling in the whiter region. Okay, and likewise, okay, so PGP minus is red and PGP plus is blue. Okay, so this is PGP minus. Okay, so because this is red here you can see, so if I zoom it, so this is the red falling in here. This is the molecule. Obviously, why molecule 1 it is showing? Because I have only attached one. If I attach multiple of them, so then multiple will fall at different places. I will show you. Let's take the example. Okay, fill it with example. You have here what do you say, three examples to be precise. Okay, three or four, yeah. So first is of brufen. Okay, I is silent over here. So brufen. So basically this is a painkiller. So omeprazole, this is for your gastritis, and this is also called as comes under your PPI, proton pump inhibitors. Okay, so likewise you can just hit on run. Okay, so once you hit on run, you can see all of them represented in one. Okay, one by one you will be having the representation. This is for the first one, brufen. Okay, so you can go by the profile of it. Okay, so I am not defining because as you can, this is self-explanatory. You can just go by it. Is it following or not following? All the reference parameters are given over here. Okay, based on that you can take the empirical decision. Okay, so all of them, as you can see, four of the compounds, as you can see, are enlisted over here. Okay, with all of their profiles. Okay, so all of the pharmacokinetic, pharmacodynamic, lyophility, then your bioavailability. Okay, then your what is the drug likeness, everything is being enlisted over here. Now let's see the boiled egg of this. So when we go to boiled egg, here you can see four molecules were there. No? So here you can see two of them are having PGP plus. Okay, whereas one is having, what do you say, like the omeprazole basically having HIA. So that obviously signifies your it has to do with your what do you say, stomach based absorption. That is why it is important. Then penetrating the blood brain barrier, brufen is capable of penetrating it, and as well as the molecule 3 is capable of doing that, and both are PGP negative. Okay, so here you can see those parameters as well. Okay, so that is about how you screen. Okay, at once also you can paste multiple what do you say, smiles, and do the screening of all of them. In CSV obviously you can download it, because going via them one by one is a cumbersome process. Obviously you will download it in the form of CSV and then see which is satisfying the parameter, which is not. For that you can also do conditional formatting. Okay, so like by changing the colors and all, and then you can do the further part of screening. Okay, so that is there. And so now let's move on to the next tool. Okay, so that is called as the ADMETlab. Okay, so we'll take our example as well as one of their example as well. So this is ADMETlab 3.0. So obviously the updated version always comes in. Okay, so if it whenever you are using, if you are using by the time the 4.0 has come or 5.0 has come, so obviously needless to say you go by the latest one. Okay, so here you have all the developmental details, what it caters to, everything is being written over here. Okay, the APIs also you can go through. Okay, so what it utilizes, okay, so it utilizes, you can see RDKit and like Django, Python, everything. Okay, so it's used for its developmental purposes. Obviously you don't have to use any of them. Okay, so it is running in the background. So you can go to the services, go to the ADMET evaluation or screening. Okay, so ADMET evaluation, let's suppose I go on to, okay, I can paste the, I already have the copied smile. Okay, so that is of your, like, this particular aspirin that you see over here. Okay, so I will hit on submit and I will see what are the parameters it comes up with. Okay, so it has much more elaborate parameters, to be honest. Okay, so that is why this is my preferred, if you ask me, this is my preferred tool to go for. Okay, instead of SwissADME. Okay, so this is latest also. SwissADME is like from many what do you say, years you are having that. Okay, so it is comparatively newer. Okay, so but you can go through the papers, multiple papers you can cite, you have, even though for the publication, if you go to, so you will see that what paper has cited this particular server. Okay, or referred this particular server. Okay, so yeah, so basically here you will be finding the basic information, that is your physiochemical properties, but obviously this particular things you can find in your normal pubchem also. Okay, so the main thing that we are concerned about is this. Okay, so QED, so the QED parameters that we were talking about in like drug likeness, here all the drug likeness parameters you can find. You can find QED. Okay, apart from that, your Lipinski's rule, Pfizer rule, GSK rule, golden triangle, pains. Okay, so all of this can be taken into account, but mostly I tell my students to refer to this five. Okay, so this five are good. Apart from that, obviously QED can also be taken into account. Okay, so all of them have their own relevance. If you just want to understand what is the Lipinski's rule, so you can go to the I button. So basically molecular weight should be less than 500 Dalton. Okay, so kilodalton, and then you have your log P should be less than 5 or less than or equal to 5. Hydrogen acceptor should be less than 10, or less than or equal to, and hydrogen donor should be less than or equal to 5. Okay, so that those are the 4 parameters. Previously it used to be 5 parameters. Okay, actually, and now one of the parameter is being removed from there. Okay, so that is the part. Okay, then you have Pfizer rule, then GSK rule. GSK rule is nothing much but just a stringenter version of this particular Lipinski's rule only, because I have already told you Lipinski's rule being the golden rule of thumb, so it is always being followed. Okay, so you want it to be followed always, and all of the parameters are derived from this. So here you can see GSK rule is nothing much, but the Lipinski's rule was giving the leverage till molecular weight of 500, it is giving till 400. Whereas the log P, Lipinski allows till 5, it allows till 4. Okay, so that much of a minute difference you can find across all of the different like parameters that you see. Okay, GSK and Pfizer are being the pharmaceutical giants. They have to like come up with more stringent parameters, because they have to get the approval from FDA and get their like what do you say, drug in the market. So that is the main thing. Okay, now coming on to your absorption parameters, so obviously all the sub parameters that you need to learn for that. Okay, so what are the sub parameters? What is kappa 2 permeability? What is MD, CK? What is PAMPA? This that you can go to I button, or I will show you a better way to like go via all of this. What are all of this? Okay, I will just show you. Okay, so before that, just focus on what is the like absorption profile, and this particular color also does matter. Okay, the color connotation over here that is represented, it is very important. Okay, so green basically signifies it passes. Okay, yellow signifies that 50-50 it is there, but obviously we mostly consider that also. Red is a no. Okay, so obviously red doesn't pass. Likewise, here you can see on wherever you see this are triple negatives. Okay, this is not not applicable. It is not that. Okay, so this is triple negative, triple positive. Okay, so don't get confused. So all of the, so obviously that particular thing also has a reference. Here you can see the reference of that. Okay, what does the triple negative mean? What does the triple positive means, or say double positive means? Okay, likewise, so basically absorption parameter, all of this. Okay, so it is not that all of them have to be satisfied. Okay, so you need to understand the importance of each parameter, sub parameter basically. Okay, under absorption, under distribution, these are the parameters that you consider under distribution. These are the parameters that you consider under metabolism. Okay, so we were seeing that their early inhibitors were there. Okay, cytochrome P53 inhibitors, but here you also get the information of substrate, that is the more thing that ADMETlab provides you. Then excretion, two important parameters are there over here. You can see T half and Cl plasma. Okay. Then coming on to your toxicity. So toxicity profile is far more elaborate in here as compared to your SwissADME. Okay, so let me get this one over here so that you can see it clearly. So here you have the toxicity profile. Okay, you can see the DILI. DILI is what? Drug induced liver injury. So how much of a liver injury it is causing? As I have stated you in the start of the video only, that the aspirin is having a profile of liver injury. So that is why here you can see it is having a red, but it is marginal. Till 0.70 it is yellow, it is 0.74, that is why. So it is not that everyone would be experiencing it, but some 10-20% or 30% of the people might experience liver injury with this particular molecule. Okay, so obviously here we are considering it as a molecule only, but although obviously it is a marketed drug already, so for past many decades. ME's toxicity has to do with your carcinogenicity again. So the carcinogenicity relates to your cancer. Okay, so obviously, let's suppose you are coming up with a drug against your any of the thing, normal cold, flu, this, that, and that is giving you cancer. Doesn't make sense, no? So obviously, so you need to check that particular thing. What is the skin sensitization profile? Okay, so then your carcinogenicity, which one is more important for your study, that you have to decide. Okay, it is not that it is like somewhere it is written that this is the absolute important and others are not important. All of them can be important depending upon what kind of research you are doing, what you are catering to, what kind of what do you say, disease you are targeting, what is your main intention is. Okay, so based on that, obviously then you have tox 21 pathway that caters to metabolomic analysis as well, but obviously not under this. So ADME are the parameters that you need to consider. You can also download the CSV from here, again do the conditional formatting. Likewise, whatever parameters are absolute important for you, you need to check for that and then do the screening further. Okay, and once the compounds which has passed all the screening of drug likeness, ADMET, everything, so now they are good to go. They are basically uh, safer to administer inside the body, and they will get onto the target. And now we will do the docking to make sense that yeah, so now it has got to the particular area where it should be binding, or where it might bind. Obviously now we don't know before docking. So then we will bind it and see that what is the affinity, how it is binding, and then it everything makes sense to you. Okay, so likewise, then you can obviously, so I was talking about how to know more about this particular, all of these parameters, because everything, covering everything in one video is obviously not possible. Okay, so like you can go to just help section over here. Okay, help endpoint explanation. Okay, help section endpoint explanation, if you go on to, okay, so yeah, so it is taking just a bit. So here you have it. So here you can see all of the parameter under drug likeness, all of the sub parameters that you see. You just click on them and you can see the what do you say, description of that, what is the empirical decision, this, that. Okay, so reference also is provided here. You can see all the references are provided. In absorption, let's suppose I want to know what is CACO2 permeability. So basically CACO2 permeability is what? So it is a human colon adenocarcinoma cell line basically. Okay, so there are multiple cell lines that are there. Again, toxicity also, you'll be finding many cell lines like your this is there. Where is it? So you just scroll down it, you will find it basically multiple of the, like you have the respiratory toxicity, this, that. Here you have heck cell line is also there, he. Oh, just give me, yeah, here you have it. Okay, so here you have all of the parameters likewise. Okay, apart from that, if you go to like tox 24, okay, in toxicity, obviously tox 24 doesn't come under, because tox 24 is a part of your metabolomic analysis actually. Okay, acute toxicity rules are there, genotoxicity is there, then you have your what do you say, oral rat toxicity, skin sensitization, how much skin rashes or blisters is that you can get. Okay, apart from that, hematotoxicity relates to blood, obviously. Okay, so then hepatotoxicity is different. Okay, hepatotoxicity is for your liver, hemato is for your blood. Okay, genotoxicity for your genes. Okay, genetic based toxicity. And here you can see result interpretation, how to interpret the results, what should be your empirical decision based on what they have marked it, demarcated it as green, yellow or red. So you can infer that why is it green, why is it yellow. Okay, so from here you can know about them. Okay, so what I was referring to, I will just let you know, just give me 1 second. So here you have the excretion parameter metabology, everything basically you can read from here. Okay, so what I was referring to you about the cell line part is this. Wait, just so here you can see HEC 293 cytotoxicity and A 549 cytotoxicity. These are cell lines. Okay, so as you understand, cell lines for different purposes. Apart from that, you can also go back and for the example's sake, I can also take the example from here. And likewise, what we did for SwissADME, it will work the same. It is just that they have their own examples. Okay, so I'm not trying to do that because it will obviously take more time, and obviously it works the same. Okay, so I have shown you using a smile. So multiple smile you can paste one after another, and it will give you, so just give a space between them and paste all of the smiles, or copy paste also will work. Okay, so copy paste all of the things, that will also work. And so here you see, let's suppose we are taking certain examples that you can take. Okay, so uh, multiple examples also you can take. Okay, so one by one the results will come, and then a series of results will come, and you can download all of them in the form of CSV and likewise. Okay, so that is there. Apart from that, one more thing is there. So if you are using any of the browsers like your Chrome or your Safari or your Mozilla Firefox, this that. Okay, in some of the browser you might get a warning while opening ADMETlab regarding privacy issues. Okay, so basically sometime what happens is the privacy policy is not being updated. So nothing to be worried about. So wherever you are getting that particular option, you just go to the advanced settings and do continue anyway. Okay, so it will be written in bracket unsafe, it would be written, but it is not unsafe at all. The security basis it is unsafe because they don't have the affirmation that the site is secure. That is why they will throw. So most of the time you won't get it, but sometimes if you are getting it, so this is the troubleshooting that I am trying to tell you. Okay, so with that particular note, so this is Pradit Banerjee signing off. So I hope you have learned something new, and in this particular video I will also attach the docking tutorial, because after this you need to pursue docking only. And as promised, so I will also try to come up with the molecular dynamics simulation in details video. And if you are new to the channel, please do consider liking, sharing and subscribing to the channel, so that you get the new videos as soon as it is published. And you can also consider clicking on the what do you say, bell icon, so that you also get the notification of it whenever a new video is released. With that particular note, bye bye, take care, have a good health.